DNA replication, also known as DNA synthesis, and it is a semi-conservative replication. DNA replication takes place in the S phase of cell cycle. Here, for the replication, origin of replication is involved. In prokaryotic cells, there is one origin of replication, that is known as origin, while in the eukaryotic cells, more than one origin of replication are present. That are sequence of nucleotides consist on adenine and thymine. Synthesis of DNA takes place in 5 to 3' direction. For the synthesis of DNA, 3 phases are involved, that are initiation phase, extension phase, and termination phase. In initiation phase, 2 enzymes are involved, that are topoisomerase and helicase. Topoisomerase is involved in the uncoiling of duplex helix, while the helicase is involved in the breaking of hydrogen bond. Here also proteins that are known as single-stranded binding proteins. This function of these proteins is to prevent the recoiling of the duplex helix after uncoiled by the topoisomerase. So look at here that is the double-stranded DNA, where the topoisomerase, also known as DNA gyrase in the prokaryotic cell, this topoisomerase come and uncoil the duplex helix. After uncoiling, this helix become just like a ladder. The SSB come here and attach to prevent the recoiling of the double helix. Then the helicase enzyme comes and start to break down the hydrogen bond between the two helix. As a result of that, a replication fork is formed. So that is the initiation phase of the DNA synthesis. After that, the elongation takes place. In the elongation phase, 2 enzymes are involved, that are DNA polymerase and primase. DNA polymerase are 3 types: DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. Their functions are: DNA polymerase 1 role in the termination, DNA polymerase 2 takes place on the repairing of damaged DNA, while the DNA polymerase 3 is involved in the synthesis of new strands. Primase enzymes that are involved in the formation of primers, which are a short sequence of RNA about 10 to 20 base pairs, and these primers act as an initiation site for the synthesis of strand. Now come toward the diagram. After initiation, the elongation started. First, the primer come here and attach to the strands of the DNA. It wants template strand. These strands then act as a template for the new strand synthesis. At one strand, one primer is attached, while at the second template strand, more than one primers are attached. Each primer has free hydroxyl group at 3' end to bind the coming nucleotides to form the new strand. Then the DNA polymerase 3 comes, which is involved in the synthesis of new strand. This DNA polymerase 3 is a huge dimer molecule. That is, it consists of 2 units that further consist of several subunits. DNA polymerase 3 cannot initiate replication process. It can add a nucleotide onto only a pre-existing 3' hydroxyl group and therefore needs a primer to attach here. That is the primer. These are the primers. This primer performs its function to initiate the polymerization process through the DNA polymerase 3. It always adds nucleotide at 3' end of the primer, so the direction of replication becomes 5 to 3'. That is, from 5 to 3' direction the DNA synthesis takes place. One of its subunit also possesses ability to remove wrong nucleotides if it is added mistakenly. This ability is called proofreading. Both units of DNA polymerase 3 are interlinked by small polypeptide chain. First unit works on one template and continually synthesize a daughter strand towards the replication fork. That is called continually growing daughter strand is known as the leading strand. That is the leading strand. While the second unit works on other template and synthesize another daughter strand away from the replication fork, as the 2 units are interlinked, so the second unit is allowed to polymerize the daughter, and the second unit that polymerizes the strand, this strand is called lagging strand, or also known as Okazaki fragments, because this lagging strand or fragments were discovered by Riji Okazaki, so name is Okazaki fragments. So that was the elongation phase where the 2 strands are formed. The last phase that is known as termination phase. In the termination phase, 2 enzymes are involved, that are DNA polymerase 1 and DNA ligase. The DNA polymerase 1 has Also, the exonuclease activity means to remove the primer, and the DNA ligase has the activity to form a phosphodiester bond between the fragments. While in the termination phase, the removal of single-stranded binding proteins also takes place. Now look here, there is the termination phase where the DNA polymerase 1 comes to perform its activity. The replacement of primer by the DNA nucleotide is carried out by the DNA polymerase 1, that is the removal of primers, and this DNA polymerase 1 has dual function. There is, besides polymerase activity, it also acts as an exonuclease. It is attached to the 3' end of Okazaki fragment where it eats DNA nucleotides so that it can extend, while on the other hand it cleaves nucleotide from the 5' end of the primer. And this way primers are removed in each Okazaki fragment is extended up to the next Okazaki fragment, but they do not join together, so they are in the form of fragments. After that, DNA ligase come, which form the phosphodiester bonds or covalent bond between these fragments to form a continuous strand. At the end, two daughter DNAs are formed, then the removal of single-stranded binding proteins takes place to recoil the two daughter DNAs to become just like the parent DNA. So that is the DNA synthesis. These are the synthesized daughter DNAs. So that is the termination phase.